When a conventional pill dissolves in the body, the dispersion is chaotic. Active ingredients drift aimlessly or clump together before they can be absorbed. Systemic delivery faces two primary physical bottlenecks: poor aqueous solubility, where compounds resist dissolving in our water-based biology, and low permeability, where they fail to cross essential tissue barriers. To compensate, doses are often increased far beyond what the target tissue requires. This floods the biological system with excess medication, triggering a massive burst-release effect and causing severe off-target toxicity. The existing bottleneck is mechanical. Modern medicine has identified effective compounds, but lacks the physical carriers to sustain them against the aggressive degradation of the human physiological environment. Solving this requires a shift away from strictly chemical solutions towards a combination of material science and targeted biochemistry. Researchers are now developing bioaerogels, the lightest processed solid materials on Earth, functionalized with mucoadhesive biopolymers to anchor them in place. By engineering these highly porous, programmable delivery systems, scientists can physically override the inherent limitations of traditional pharmacology. An aerogel is defined as a three-dimensional polymer network, where the internal liquid has been entirely replaced by a gas. This leaves behind a structural skeleton with over 90% empty space, forming an interconnected open-pore network that operates at the mesoscale. These pores measure between 2 and 50 nanometers across. Because the network is so dense with microscopic voids, a single gram of this material contains over 200 square meters of internal surface area. This geometry dictates the behavior of the loaded drug. The massive surface area prevents the therapeutic compound from forming its standard crystalline structure, forcing it into a disorganized, amorphous state across the internal matrix. In this amorphous state, the drug is highly exposed. This maximizes the contact area with aqueous biological fluids, which significantly increases the rate of diffusion. This contrasts with traditional solid pills, which dissolve slowly and inefficiently from the outside in. The aerogel overcomes the solubility problem through the mechanical manipulation of surface area. Producing a matrix that is 90% empty space introduces a difficult mechanical challenge: how to remove liquid without crushing the skeleton. The physical geometry that solves the solubility problem also makes the architecture highly vulnerable to the thermodynamics of traditional manufacturing. Every aerogel begins as a liquid-filled hydrogel. Removing that liquid while maintaining the void is a structurally violent process. During standard evaporation, the retreating liquid generates surface tension. These adhesive forces pull on the delicate polymer walls, causing the microscopic pores to cave in and the structure to collapse. Freeze-drying is similarly destructive. As water freezes, it expands into ice crystals that fracture the fragile polymer network from the inside. The primary threat to structural integrity is the phase boundary, the transition line where a liquid becomes a gas or a solid. To bypass this, engineers utilize supercritical carbon dioxide processing. This phase diagram shows the states of matter based on temperature and pressure. By pushing carbon dioxide past its critical point, it enters a supercritical state, where the distinct boundary between liquid and gas disappears. In this state, the supercritical fluid can circulate through the hydrogel, replacing the liquid solvent without exerting any surface tension. When the system is depressurized, the CO2 vents away as a gas, leaving the three-dimensional network completely empty and structurally intact. This process also allows researchers to utilize poorly water-soluble materials that would normally be unsuitable as standard hydrogel components. Supercritical processing provides the mechanical workaround to build the carrier, but the system needs a way to anchor to a biological target. To localize the drug, we shift focus from the carrier's internal thermodynamics to its external destination, the mucous membranes of the respiratory and digestive tracts. These internal surfaces are lined with mucus, which is primarily composed of water and a protein backbone called mucin. Mucin features an extended, highly complex brush-like structure. It is composed of high molecular mass glycoconjugates, intricately linked to numerous carbohydrate chains. To attach to this surface, engineers use alginate, a naturally occurring anionic polysaccharide derived from brown algae. By fabricating or coating the aerogel skeleton with hydrophilic alginate, the carrier can bind directly to the brush-like mucosal surfaces. This mucoadhesion grounds the delivery system, prolonging its residence time at the target site. This allows for selective absorption and enables the payload to bypass hepatic first-pass metabolism. The final challenge is protecting the aerogel from premature destruction, particularly in the highly acidic environment of the stomach. Researchers have tested this using hybrid silica and alginate aerogels loaded with ibuprofen via supercritical adsorption. To shield the carrier, they applied a copolymer coating known as Udroget L, which makes the formulation enteric and pH responsive. This graph tracks drug release in simulated gastric conditions. At a pH of 1.2, the enteric coating remains stable. Less than 20% of the ibuprofen is released over a 2-hour period in these acidic conditions. However, as the carrier moves from the stomach into the simulated intestinal environment, the pH levels shift toward neutral. When the pH reaches 7.2, the coating dissolves. This instantly exposes the aerogel matrix and triggers a vertical spike in release, deploying the full payload in under 10 minutes. Combining the aerogel matrix with a responsive coating creates a programmable trigger. The payload is heavily shielded during transit and deployed only when it reaches the specified biological target. The result is a multi-layered microapparatus, a high-capacity aerogel architecture produced through supercritical manufacturing and designed for localized mucosal targeting. This multi-stage physical construction allows researchers to bypass the low solubility and poor permeability that often limit conventional pharmacology. It enables advanced oral, nasal, and pulmonary delivery for complex therapeutics, including medications for cancer and heart failure. While early toxicological studies indicate these silica and polysaccharide systems are safe, they remain largely untested against the full complexity of the human biome. The next frontier for researchers involves extensive in vivo validation against the mechanical forces, natural surfactants, and enzymatic stresses found in the digestive tract. The future of advanced medicine relies on treating drug delivery as an engineering discipline. Efficacy is increasingly dictated by the precise physical structures that carry a molecule into the body.